Được nha.
Cảm
IBSG xin
ơn.
được gửi lời chào tới tất cả các anh chị bạn em. Cảm ơn anh chị bạn em đã dành thời gian để join vào IBSG Webinar tháng 4 năm 2025 ngày hôm nay của chúng ta. Và trước khi chúng ta đến với IBSG Webinar thì em xin được mời anh Vũ giới thiệu về một số chương trình sắp tới của IBSG nói chung. Em mời anh Vũ.
Cảm ơn Bích Nụ. À, Trong chương trình của IBC của tháng tư này thì vào cuối tháng tư chúng ta sẽ có một buổi Journal Club của nhóm của em Võ Châu Duy Tâm. Thì Tâm hiện thời đang làm postdoc bên MIT và sẽ hướng dẫn các em đọc một bài của lab em ấy, lab của thầy Phương Gen về một cái khám phá rất là mới, đó là về cái protein fencer. Nó được xem như là một cái hệ thống CRISPR-Cas9 trong tế bào nhân thực. Thì đây là một cái khám phá rất là quan trọng và được đăng trên tạp chí Science. Và báo hứa hẹn sẽ là một cái buổi thảo luận journal group rất là hay, bởi vì chính từ những người khám phá ra nó sẽ thảo luận về bài báo đó, cho nên là rất là mong các anh chị em sẽ lên nghe. Và đến tháng 5 thì chúng ta sẽ có một buổi webinar của một bạn đang làm nghiên cứu sinh tiến sĩ tại Đại học National University of Singapore, bạn Trần Minh Phương Nam. Bạn là một superstar ở Đại học Quốc tế, Đại học khoa học tự nhiên ở Đại học Quốc tế, Đại học Quốc gia Thành phố Hồ Chí Minh. Và hiện thời bạn đang ở năm cuối của PhD, và bạn sẽ thuyết trình về cái công trình mới với xuất bản của bạn về biomaterials. Thì đó là hai hoạt động. Và đến khoảng cuối tháng 5 thì chúng ta sẽ có buổi Journal Club cuối cùng của năm, của nửa đầu năm 2025, của nhóm của Tô Văn và Lauren Đặng. Cho nên là rất là mong các anh chị em sẽ theo dõi lịch trên webinar, trên Facebook và trên website để cập nhật và tham gia ha. Còn sau đây thì mình sẽ nhường lại sân khấu để cho Bích Nụ.
Cảm ơn anh Vũ đã chia sẻ rất là chi tiết về ba event sắp tới trong tháng 5 và tháng 6 của chúng ta. Và để anh chị bạn em không cần phải nhớ chi tiết về những buổi như vậy thì em có gửi vào trong phần chat box thông tin của website, Facebook và YouTube của IBSG. Mọi người có thể follow và cập nhật thông tin tin tức mới nhất. Và nhất là trong tháng 5 này chúng ta có Journal Club về CRISPR-Cas9 là một kỹ thuật rất là hữu dụng và phổ biến hiện nay rồi, thì mọi người có thể cho nghe và trao đổi chi tiết hơn về chủ đề topic mà mọi người sẽ làm về protein trong tháng 5 này. Và ngay sau đây chúng ta sẽ quay trở lại với IBSG Webinar. Trước tiên thì IBSG Webinar xin được gửi lời cảm ơn tới chị Lê Huyền Diệu Linh. Cảm ơn chị rất nhiều vì đã dành thời gian Easter và thời gian mà đang được free để lên với IBSG Webinar ngày hôm nay. Và ngay sau đây em xin phép được giới thiệu một đôi chút thông tin về chị Lê Huyền Diệu Linh. Chị Lê Huyền Diệu Linh tốt nghiệp cử nhân ngành sinh học tại Truma State University và nhận bằng tiến sĩ chuyên ngành khoa học thần kinh tại University of Rochester School of Medicine and Dentistry năm 2023. Trong giai đoạn PhD, chị Diệu Linh là một nghiên cứu sinh rất là productive. Chị đã xuất bản rất nhiều công trình với vai trò là tác giả chính trên tạp chí Scientific Report và Cell Reports. Ngoài ra, chị Diệu Linh còn xuất bản 5 bài báo cáo với vai trò là đồng tác giả và một chương sách với vai trò là tác giả chính. Bên cạnh nghiên cứu khoa học rất là xuất sắc thì chị Diệu Linh còn là một mentor hiệu quả và được vinh danh với rất nhiều giải thưởng, bao gồm giải thưởng Edward Pitt Curtis dành cho sinh viên cao học xuất sắc và giải thưởng Outstanding Student Mentor Award. Và hơn thế nữa, chị Diệu Linh còn là một người giao tiếp trong khoa học rất là tuyệt vời. Điều đó được khẳng định thông qua những giải thưởng danh giá của Đại học Rochester dành cho sinh viên cao học, bao gồm Wallace Owen Dissertation Award, Doty Award Outstanding Dissertation in Neuroscience. Và hiện tại chị Diệu Linh đang là một nhà khoa học thần kinh tại tập đoàn y khoa SetPoint. Và đến với chúng ta ngày hôm nay, chị Diệu Linh sẽ mang tới một chủ đề vô cùng là thú vị và mới về chủ đề liên quan đến adult minds. Và không để chúng ta phải chờ đợi lâu hơn nữa, ngay sau đây em sẽ xin phép được nhường lời tới chị Diệu Linh. Và trong quá trình chị Linh đang trình bày thì rất mong là anh chị bạn em chúng ta sẽ không bật mic để tránh các âm thanh nền. Và sau khi chị chị Diệu Linh hoàn thành phần trình bày thì chúng ta sẽ tới với phần Q&A. Đồng hành với chúng ta ở phần Q&A sẽ là anh DrLê Huy Vũ. Anh Vũ sẽ giúp mọi người bật mic để mọi người có thể giao lưu đặt câu hỏi trực tiếp với cả chị Linh. Nên là trong quá trình chị Linh trình bày mọi người có thể đưa câu hỏi về phần chat box kèm với tên tiếng Việt có dấu của mình để cuối chương trình anh Vũ sẽ gọi tên và mời mọi người có những phần trao đổi chia sẻ trực tiếp với chị Linh ạ. Em xin cảm ơn chị Linh và ngay sau đây em xin được mời chị Linh share screen về phần PowerPoint trình bày của mình.
Cảm ơn Nụ nha, để mình share screen. Em thấy không bé ơi?
Dạ em thấy rồi. Em xin mời
Ok
chị ạ.
ok. Mình xin phép được nói tiếng Anh nhé, tại vì là tiếng Việt thì chắc là ngồi đây đến sáng mai mất. Thì cái talk của mình based on a paper that actually had just been published in Cell Report earlier this year. Is on the microglia response to inhibition of colony stimulating factor 1 receptor differs by sex in adult mice. It sounds like a mouthful, but is the story is also a little bit complicated, but I'll walk you guys through it as we go on. So first of all, let's just go through to ask what is this one? Okay. What is microglia? So we're just gonna go through very quickly microglia ontogeny and basically what does it do? Microglia is often referred to as the um, resident macrophage of the brain. It does arise from an uncommitted embryonic myeloid progenitor cells during embryonic day 7 to day 7.5. And then it slowly travels to the brain and find it homing in the brain. The signal is dependent first on Pu1 and RUNS1, like a lot of the other myeloid cell lineage, but then it's dependent on F8, RUNS1, and then the homing to the brain is highly dependent on CSF1, and then it's maintained its population. The homeostasis of microglia is dependent on TGF beta 1. So microglia enter the brain before the blood-brain barrier form. The blood-brain barrier form usually during embryonic day 14. The microglia already make it home to the brain around embryonic day 9 and then is clonally expand. So it doesn't actually have new population entering the brain often. They just self-replenishing themselves from the population that's already entered the brain during the embryonic day 7 to 9 in development. Microglia, once they in the brain, is so they are immune cells. They actively surveil the brain parenchyma. Usually, the homeostatic microglia would adopt the structure of a ramified processes with a small cell body, but microglia are extremely dynamic cells. They can change in their morphology, which will dictate their functions. So once they become activated, they always active, but once they become activated in the presence of pathogens, they can change their shape and has this function. So the processes become thicker and they retract their processes more. Once they adopt a more phagocytic morphology, which basically become very blobby. They can start like eating and digesting the pathogens and other debris in the brain. Of course, because they're dynamic, they can always go back to their original homeostasis form, which not always guaranteed, but hopefully in a situation where you have an acute injury, microglia will safely go back to where it originally is after it's cleared out the debris from the injuries. This is a video collected by a senior grad student in the Microglia lab where I work in my PhD. So what you're seeing right here are microglia in the mm, microglia in the brain of a mouse. The green depict. Để mình bỏ cái này đi thử. Tại sao nó lại bị ý nhá? The green depict green fluorescence protein. So we are looking directly onto the microglia from a two photon microscope. So this is just brain of a mouse that the microglia are labeled with GFP. You can see that it's dynamically surveilling and patrolling the environment at all times. So they're not static cells, which they originally thought to be, but they are extremely dynamic, always monitoring their environment in the presence of a very acute injury. So you will see this blob light up pretty soon, like right now, which is a laser injury that get caused by getting laser pulse at a single point in the brain of this mouse at a very high dose intensity. And then you can see that the microglia start sending their processes, so they're swooping in and try to clear up the debris caused by this laser injury. Let's see. Okay. Uh, but microglia in the brain, once they establish themselves relying on CIF1R signaling to survive, in the presence of CSF1R antagonist, the microglia will be depleted. So the lack of a survival signal will cause microglia to undergo apoptosis. And as you see here in this schematic, almost all the microglia actually got wiped out of the brains in these mice. These are young, about 3 months old mice. Almost all the microglia will get digested by actually the microglia that remain. So microglia, because they are the main immune cells of the brain, they also the main phagocytic cells of the brain. They are in charge of clearing out the microglia that actually get apoptotics from receiving CSF1R antagonist. And once these microglia eating the microglia that got affected, the astrocyte will actually come in, swoop in, and then get rid of these microglia as well. These drugs start to get really attract a lot of attentions because they start to be used in a lot of research concerning microglia survival and in a neurodegenerative disease when the microglia became chronically active and you want to get rid of them to reset the brain environment. On the Additionally, CSF1R is also the signal for a lot of the myeloid lineage in the periphery. So they also get used in some cancer context in both research and in clinical settings as well. So understanding how microglia responds to CSF1R signaling is crucial in developing this drug to be used in humans for altering disease states in neurodegenerative diseases. In aging and disease, microglia significantly downregulate their CSF1R receptor expression. So as you can see in this figure, when they're really young, when they're about 2 months old, which is when people most often test these drugs, because you don't want to wait unless you really study aging, right? So people usually just test the drug in really young mice, which they have a really high amount of CSF1R, but the level of CSF1R gene expression drop dramatically as the mouse age, and once it gets to like a year old, you have very little of this gene left. And what we've seen in our lab, and others actually start to report, is that once the mice get really old, so this is 22 months old mice that have a triple transgenics for developing plaques and tau pathology for replicating Alzheimer's disease pathology. So here you can see in red, 6E10 is just a marker for amyloid beta plaque in this model. ABA1 is a marker for microglia, and this is just a merged image. What you can appreciate right away is that in the control mice, you see a lot of microglia present. But when you feed the animal with PLX, which is a CSF1R inhibitor drug, you can see the microglia going away in decent amount, but this is not what you see with young mice. So in the next few slides, you'll see in the young mice, it's almost gone. But in this age mice, there's only about 50% of the microglia that get reduced in distal from the amyloid beta plaques, but closer to the pathology itself, the microglia proved to be extremely resistant to being depleted by this drug. So, which means that these microglia start to employ different survival mechanisms. And if we want to use this drug to treat, for example, Alzheimer's disease, we want to have to think about what dosage we want to use. Do we have to alter their pharmacokinetics so that it penetrates the brain more? Or is there any way we can make this microglia more susceptible to be depleted by this drug? So my questions is, are there sex-dependent differences in their response to CSF1R? And if so, are there underlying biological mechanisms that give rise to such differences? this is when we see a lot of, like sex dimorphism in disease prevalence. For example, Alzheimer's disease affect women way more than men, but Parkinson affect men way more than women. So can we learn something about how these cells respond to these specific drugs and how they differ between sex? Are there any basal differences in them, and what happened when they under the effects of CSF1R inhibitors? So what I see firsthand, so these are very serendipitous findings. I was actually trying to use this drug for a different project and found out that I just cannot deplete the microglia in female brains. So I try with younger animals, but I still find that the microglia remain surprisingly very resistant to be depleted by this PLX drug. Here I'm using PLX 3397, which is CSF1R inhibitors, but it is a little bit non-specific, so it's also depleted some oligodendrocytes. As you can clearly see in the control brain, this is again IBA1 for microglia. There are a lot of them present. They about 5% of the cell, 5 to 10% of the cells in the brain are microglia. So you see a lot of them present. But under the treatment of PLX, the male microglia are almost gone. So they wiped out about 80% of the male microglia versus the female, there's still a substantial amount of them left. And that data is quantified here. So the female are in the white hollow circles and the males are just the filled green circles. What you can appreciate is that there are a striking difference in the depletion density the depletion versus control density ratio between female and male, the ratio closer to zero would indicate that the microglia completely get wiped out, and the ratio closer to one would indicate that there are not a lot of differences between when control and depleted brains. While in the female, we still have, like about 50 to 60% of depletion. In males, there are more about like 80% of depletion across the board in different brain regions that I assay. These microglia, as a result, because they are remaining and they have to eat the carcasses, literally carcasses of the apoptotic microglia, they become a little bit more activated, and you can appreciate that their soma size got a little bit bigger, so the ratio between depletion versus control is slightly bigger than one, indicating that their somas is slightly bigger. It's not significant difference between male and female though. So next, what I attempted to do was doing a morphology analysis of the microglia between control and PLX treatment. There is a very well-established method to quantify microglia morphology, which is show analysis, in which there is concentric circles rings that are radiated out from the microglia soma. So it will be like a circle placed in the soma, and then there's just the same constant intervals between the rings, and they would just count the number of times that the dendrites touches these rings, and the maximum number of the dendrites touching these rings would indicate that the microglia morphology is ramified and complex. Of course, there's also total intersections, MAC intersection, and the size of the actual circle itself. But to simplify things, let's just look at total intersections. So just quantify grossly if microglia reduce in their ramification at all, irregardless of whether the process actually got smaller or more shorter. So we can see that in both male and female, the microglia after PLX treatment got significantly more activated. So their total intersections with these concentric circles get significantly reduced. So if we go back just a few slides ago, I'm sorry, this is not very ideal. They would look more like this coming from this structure. They would reduce their ramification, the process got thickened, and the soma actually got a little bit bigger. So to return to our story, we then ask if this change is because there is a change in how PLX got metabolized. And so we increase the dose of PLX by about 40%. The reason we choose 1.4 times of the normal dose is because we noticed that the male got depleted about 1.4 to 1.5 more than female. So 20 to 30% depletion compared to 40 to 50% depletion in the female. So the line of thought is if we just linearly increase PLX, will we see a difference? And of course, if we just hit it with a bigger hammer, the female also decrease in their microglia in response to higher doses of PLX. While the male, the microglia almost got entirely wiped out. There's very, very few of them left. But surprisingly, this change in the level of depletion is not correlated to the brain concentration of PLX. This is notwithstanding that we only do one time point across the board. So we just sack the animal in the morning and then just ship the brain out for concentration quantification. There need to be a more systemic way of doing this. You ideally want at least like 3 time points during a day to make it worthwhile doing this experiment again. And then we ideally would time the estrous cycle of the female as well. But this is already show us that the PLX get into the brain of these male and female mice just fine. So it's not like the female dramatically just don't let this PLX get into their brain. So next we want to know if they just generally eat less. So it is well known that male and female mice eat approximately similar amount when they in adulthood, with the male eating a little bit more, and this is kind of what we see here as well. So consistent with the theme, female is always in white circles and the males are again in this green circle. We see that there's no change, no correlation between weight of the mouse and the total food they consumed. Furthermore, when we look at the amount of food consumed versus the number of microglia per millimeter square in cortex, we see that despite eating the same amount, the number of microglia is so much higher, that remain is so much higher in female compared to male, and that is also true in the hippocampus and other brain regions, but the number is not shown here. So this tells us that there's something inherent difference about these microglia between sexes. So it's not just that female microglia uh, it's not just that female mice just don't have PLX in their brain. It's not that they're eating less PLX. It's more like at baseline, this male versus female microglia utilize different survival mechanisms, and maybe the female is just way more resistant to being depleted by CSF1R inhibitors than male, suggesting that they might utilize different survival mechanisms in addition to the ones that utilized by both sexes. So to answer that questions, we turn to flow cytometry and bulk RNA sequencing analysis. And for this experiment, again, using very young mice, just to make sure that I can actually get the microglia to be gone, feed them with PLX, the CSF1R inhibitor, or control food, the child diet, and then I dissect out the cortex and hippocampus, and then sort out the microglia. I get and sort them out, and then I do bulk sequencing. So there are a couple of proteins that worth mentioning that are actually interesting, and we focus on which the first two are P2Y12 and TMAM119. So as I mentioned before, in the homeostatic brain, the microglia are extremely ramified. And to maintain this structure, there are several key homeostatic proteins that are upregulated in a normal situation that these microglia are dependent on. Two of the most prominent ones are P2O12 and TMA119, which is expressed really high in adult homeostatic microglia. Once the microglia became activated like this, so if they look blobby and start to carry out a lot of the phagocytic functions, the homeostatic genes get dramatically downregulated, and lysosomal CD68 starts to increase as we all know for the microglia to start ingesting and phagocytosed debris. So the microglia get gated first on just singlets, and then they get gated on 7AAD, which is the lydate marker, so I only choose lye cells, and then they gated on CD11B positive, CD45 intermediate as a generic marker for microglia. And then the intensity of a lot of proteins get looked at, but prominently these three are the most interesting one in this experiment. And then the same microglia also get sequenced. They get sorted out and then they get sample sequencings. And from this PCA plot, you can see that the female microglia and the male microglia, so account for the account for the smaller differences in these microglia. So this is PC2, and as you can appreciate, the female control in this lower right corner and the male control are pretty similar. The male and female PLX are slightly different. The component that account for the most difference in clustering these groups is actually just PLX treatment, which is not surprising because we basically say putting a drug in that try to kill it, and then only the ones that are the most non-resistant to the drug that got survived. So CSF1R in general, both sexes upregulate activated marker and downregulate homeostatic signatures in microglia. So this is just, first of all, confirming our data from immunofluorescence analysis. So we see that the number of cells that's still alive in female after 3 days or 7 days of treatment, they are decreased, but not significant. This is just because there's some that just have very little microglia that actually survive the FACS sorting process, which is quite stressful. But in the male, you can see that the group are clustered very close together, and the microglia decrease in a graded manner, which is what we expected. For the P2H2 expression, which is one of the homeostatic marker, it decreased in both male and female as expected, as same as the TMEM, or another homeostatic marker, and the marker for activation increased in both male and female. So we do expect a lot of similarity in these microglia between sexes because after all, they are getting activated by the drug and they have to get rid of all the microglia that are undergoing apoptosis after the drug treatment. And then this is just showing the very similar upregulated pathways in male and females in upregulating a lot of their immune response pathways. But what is more interesting and what we're really trying to focus on is the sex-dependent differences in these microglia. At baseline, there are very limited sex-dependent differences in the transcriptomic signature between male and female. A lot of the genes that are different between male and female microglia are actually just X-linked genes. So they are just inherently going to be difference between sexes. So DDX3Y is one of them, UTI is one of them, KDM5D is another one. So the most significantly regulated genes are just genes related to sex-related genes itself. But there are some differentiated genes that also get reported by other papers that suggest that at baseline, so when we look at the GO pathways upregulated in males, there's some upregulation in chronic inflammatory responses, especially NF-kappa B signaling pathways that get upregulated in males compared to female already at baseline. Not to a great extent, but they are at a little bit of a elevated activated levels at baseline. But these difference in sex got pronounced when the mice are treated with PLX. So you can appreciate from maybe this plot, there are way more genes that get differentiated after PLX treatment. And the pathway that I want to highlight in this GO analysis is that while the female upregulate a lot of pathway related to autophagy and mitochondria maintenance, a lot of the pathway that upregulate in males have to do with electron transport chains subunits. So a lot of the genes in subunit ones, half of them got upregulated in males after PLX. And they also upregulated mitobiogenesis. So not necessarily mitochondria surveillance and dynamics, but more just making more of the mitochondria, which we found really interesting and what we pursue further with this paper. But before we get there, let's just take a step back and talk about the microglia that actually surviving the treatment with an inhibitor that basically say kill yourself. These microglia, how are they different from the other microglia, and why do we see a graded response? because we do know that male and female are different, and then when we increase the amount of PLX, we also see a graded response. So what are the differences that these microglia have? There's a paper published in 2020 that already show that there's a population of microglia, galectin-3 positive, or in this paper they call it MAC2 positive, which is the same protein. The microglia again is represented by this red color with IBA1 labeling, and MAC2 is in green, so any overlap would show up as tilted yellow. DAPI is just a nuclear stain in blue. So you can appreciate that in control animal, there are very, very few microglia that actually express MAC2 or galectin-3 positive, compared to the total number of microglia. I want to make that very clear because we will see the difference later. So proportional wise, you see like one positive cells among a bunch of cells that get captured in the image versus in day zero, so immediately at the termination of the drug. You can clearly see that the microglia now express really high level of galectin-3, and the ones that survive all express galectin-3. In this particular paper, except like this one. In this field view, there are 2 out of 3 that express MAC2. So this is the data they quantify. So if you count the absolute number of cells, there are way more microglia in control versus immediately after termination of the drug, not surprising. Which IBA1 positive, MAC2 negative, which almost all microglia. The population of microglia that also express galectin-3 actually doesn't change that much after PLX. There's a slight increase and it's significant, but it's not major. The things that actually differ the most is that the microglia that are not expressing MAC2, or galectin-3, the IBA1 plus MAC2 negative population almost got completely wiped out, while the population of microglia that actually express galectin-3 actually surviving the treatment with CSF1R inhibition. So the difference between the proportion and the number of microglia is significant because it signifies that these microglia not just arise from nowhere. These microglia that surviving the treatment come from the microglia from the control brain that's already expressed some galectin-3 that got either upregulated or maintained in their level as the other microglia got depleted. And this is also what we kind of observe as well. So to replicate that finding, we also look at microglia staining here in green, galectin-3 in purple. Any overlap you would see as white. So in the control brain, the upper panels are females, the lower panels are males. You can see that there are very, very few cells in proportion to the whole field of microglia that actually express galectin-3 or MAC2. For example, in females, there are like 1, 2, 3, 4, 5. In males, there's only 2 out of hundreds of cells in the field view. In the PLX treatment, however, almost all of the remaining cells in males expressing some level of galectin-3, and there are some in females that also express galectin-3. What is surprising for us, this is based on the high variability, there's not significant difference, but it appears that female at baseline seems to express slightly higher percentage of microglia that co-express galectin-3. Maybe suggesting that because of this expression of galectin-3 at baseline, that is why the female microglia are a little bit more resistant to being depleted by CSF1R because they already have more of the populations are resistant to being depleted by the drug. Because we see a difference in the expression of galectin-3, we classify this by high expression versus low expression, but for the simplicity, they all express galectin-3. So we can just look at this graph as whole, but in the paper, I do differentiate between low versus high expression. What you can see is that the percentage of IBA1 cells, so just microglia, that are galectin-3 positive is a lot higher, so it's dramatically increased after PLX in both male and females, but there are higher percentage of microglia that expressing galectin in male compared to female. And that is not true of the absolute number of cell counts. So the absolute number of cell counts also increase with PLX, but to a less extent. So here you see almost no 0% or just like 1 to 2% of microglia that actually express the drug. But when you count the absolute number, the increase is much less pronounced, as previously observed, because these microglia actually get resistance. Of course, there are other microglia that get upregulated in their expression of galactan-3 that lead to a slightly increase in the number of microglia that co-express galactan-3. What is interesting is that while the absolute number of galactan-3 positive microglia is the same between the two sexes, because there are way more microglia that remain resistant to the drug, the percentage of microglia that express galactan-3 in female after PLX is lower compared to male. So we also want to ask, okay, if male and female depleted microglia to the same extent, which if you remember a lot of slides ago, I present data on a slightly higher amount, a slightly higher dose of PLX. With 1.4 dose of PLX, the female microglia depleted to the same extent as the PLX male microglia. So now we have similar depletion rate. So with the same number or amount of microglia that remain after the drug treatment, will we see a difference in how these microglia behave? And the answer is yes. So we see that even when the microglia got depleted to the same extent in male and female, the female microglia still express less galactan-3 positive cells compared to male, which suggests to us that because of these differences in how they express galactan-3, again, they might be inherently employing different survival mechanisms. It's not just that they are, not getting the drugs or if they're not eating enough. It's really more something to do with their intrinsic difference in how they govern their survival pathways. And I also analyzed the hippocampus in addition to the cortex with the same stories. Uh, So next we look at the expression patterns of these galactan-3 positive cells at baseline and also under CSF1R inhibition to see if there are any pronounced difference in how they regulate their survival pathways. And for this, again, I employ flow cytometry. These mice are again treated with PLX, the CSF1R inhibition versus control, and then their cortex get dissected and the microglia get tagged with a bunch of proteins. And then I look at the protein's expression. So similar to what we see with immunofluorescence analysis, there are significant increase in the percentage of galactan-3 positive microglia in both sexes with more pronounced increase in male. So it's good to have confirmation. The other thing that we find that is quite interesting is that not only the male upregulate more microglia that have galactan-3, the intensity of galactan-3 expression in male microglia is also higher when compared to female. So from this point forward, the PLX is now in blue and the control is in white, just to make it consistent, because there are going to be some shifting in how I name the y-axis. So again, the microglia got selected based on their traditional markers, CD11B positive, CD45 intermediate, and then they get gated for galactan-3 positive versus galactan-3 negative, which was done on some, for instance, minus ones controls. And then they get grafted, and then their intensity get computed. What is the most surprising to us? So now here we go back to the mitochondria story that I said earlier that was interesting that the female upregulate mitochondria surveillance and dynamics and maintenance, so basically autophagy pathway, versus male upregulated a lot of complex ones, subunits, genes, as well as just mitochondria biogenesis in general. Here we see that galactan-3 positive cells in female, the upper panels are all female, already upregulated mitochondrial which is the proxy for mitochondria mass compared to galactan-3 negative cells, which might suggest that even at baselines, these galectin-3 positive cells already upregulated their mitochondrial biogenesis, which favor toward proliferation and maintenance of these cells. So maybe that is why they are more resistant to being depleted and survived after the drug treatment. In addition, these galectin-3 positive cells surprisingly also upregulated CSF1R receptors. So at baseline, they already employ more receptor for the survival signal. They, in addition, also upregulated TREM2, which is a receptor that acts through adaptive protein with beta-catenin downstream to increase survival and proliferation signal. So at baseline, the galectin-3 positive microglia upregulated multiple survival signal paths, suggesting that they are primed themselves to be very resistant to get hit by inhibition to any of the survival signal they have. The same story go to males, because I place them kind of like in this way, it's hard to see, but you can appreciate that in male, there's similar story. The galectin-3 positive cells also express higher level of mitochondrial which a proxy for mitochondria mass. Um, they also express higher level of CSF1R. Here is the control in white. And then they also express higher level of TREM2, also a control in white. I hope what you can appreciate is that when we compare male versus female, what happened is that after CSF1R inhibition, male really downregulate their CSF1R expression, so they go it is like a fourfold differences in how they downregulated their protein levels after CSF1R inhibition, so after PLX drug, which is shown here in blue, versus in female, there is a much milder decrease in the level of the receptors, even with the inhibitors around, suggesting that these female are there might be some inherent difference in how they regulate their receptor dynamics to respond to the drug that beyond the scope of my study, but is interesting. So next, we want to compare male versus female and the differences in how they express these markers. So here again, the white empty circles are still control and the blue field circle are still PLX, but now the y-axis become male versus female. So these are all microglia together, both galectin-3 positive and negative cells. So for all microglia, we see that yes, both male and female upregulated their mitochondrial mass. Remember that these cells are now activated, so they just have to exert a lot more energy. But what is striking is that male express way higher level of mitochondrial compared to female. So they in agreement with our RNA box sequencing data, male do upregulated mitochondrial biogenesis a lot more, and because they also express a lot of the gene related to electron transport chain complex 1, which is known to regulate the RES pathway, so the reverse electron transport chain pathway that lead to inflammations and cell death. So we hypothesize that because they express way higher mitobiogenesis as well as complex 1 subunits genes, it might prime themselves to just clear out the apoptotic cells way faster when compared to female. Another striking finding that I already alluded to in the earlier slide is that while both male and female downregulated their CSF1R receptor expression, the degree to which microglia downregulated the receptor expression is way higher in male versus female, suggesting that there still might be enough CSF1R receptor around for the female microglia to maintain a decent population. We also look at the ligands alone, so not the receptor, but CSF1 ligands, and found that the CSF1 ligands increase in both male and female in a probably an autocrine manner when you're just trying to hit it with a hammer and inhibit the receptors, the ligands, the endogenous ligand is gonna the autocrine regulation is going to make it increase in expression a little bit. What is also surprising for us is that there is a slight difference in how male and female upregulate their TREM2 expression after being inhibited by CSF1R inhibitors. So with the PLX treatment, the male microglia express slightly higher level of TREM2 compared to female, suggesting that female might not utilize the TREM2 survival pathway as much as male under CSF1R inhibition, which might make sense because they still have a lot of the receptor left compared to their male partners. So next we are interested in asking, okay, so if these microglia try to utilize different pathways so they can survive, can we try to kill more of them, basically? So this is a very practical question because these drugs start to get a lot of attention in Alzheimer's disease. So trying to use CSF1R inhibitors, the PLX drugs, to get rid of microglia, to reestablish the niche, to make sure that you get rid of a lot of the chronic inflammation. So the question really is, can we just clear all the microglia? So to address this question, we first actually thought of just using a TREM2 knockout. So the line of thought is that, okay, we just knock out TREM2, which is another survival pathway that these microglia employ. Can we just kill a lot more of them? As you can probably guess, that's not as easy as that. So there's a lot, it's very busy, but again, the upper panels are female, the lower panels are male, and I want to shift your focus on the first bar and the second to last bar. So the first bar are control. You can see that again in control brain, female get depleted less than male. There are about 50% of the microglia left versus in male, there's only about 25 to 30% of microglia that's left after the PLX treatment. In TREM2 knockout, which we expect is that we'll see microglia all gone. Instead, we see even more of them survive after the treatment, especially in female. So there are a lot more microglia just left after the treatment. There's no differences compared to control. When we divide it by region, there are slight differences in some region, but all toward increasing their survival rate after depletion. So we wonder, well, why that is the case? Because we thought that if we just inhibit another survival mechanism and then hit it with a hammer for CSF1R, basically, we'll get rid of all of them. But it is another well-known fact in the field of Alzheimer's disease that TREM2 knockout actually lock microglia in their homeostasis state. So with TREM2 knockout, these microglia cannot get to their activated state, so they don't turn blobby. They stay in their homeostatic state. And they actually don't proliferate to encapsulate the plaques, or if there is an injury, they actually don't mobilize to the injury site as fast as in the TREM2 control mice. So the line of thought became, okay, so what if the baseline activity of microglia has something to do with how they get depleted? To address this question, we turn to another mouse model that are very commonly used that is also well known to alter microglia basal state, which is CXCR1 or fractalkine knockout mice. So knocking out of fractalkine is well known to lead microglia to a more activated state, with the haplodeficiency be somewhere in the middle. So they're known to kind of initiate this premature aging phenotype of microglia. So they start to get more activated, they become more quote unquote more senescent and more quote unquote primed for being activated. And that data is represented here in the darker orange bars. So as you can appreciate really fast, is that when we completely knocked out CX3CR1, as I shorten as CX3, the female microglia got depleted to the same extent as the male. So most of them are gone. Versus the CX3 heterozygous, which is the haplodeficiency of this fractalkine receptor, the microglia depletion kind of got somewhere in the middle. So we see a very well graded effect of depletion of microglia when as the microglia activation state slightly getting higher. Surprisingly, we don't see this effect in males. So only in females, when we induce this change in the baseline activated state of microglia, that we see a correspondent change in how they get depleted by the PLX drug. Um, which kind of come full circle to our thought that there are inherent difference in their baseline activated states, and that's what lead to the changes in response to being depleted by PLX. So if you can recall that I said male and female microglia at baseline look very, very similar, but the male microglia upregulated NF-kappa B signaling pathways and some of the chronic immune modulation pathways. So they're a little bit more activated just at baseline. And we thought that maybe that is what driven them to be quote unquote killed quicker by PLX drugs. Another surprising findings is that if we combine CX3 heterozygous with TREM2 knockout, so not just knocking out the TREM2, not just chucking one of the survival mechanism, but if we also coupled it with a lack of a copy of this fractalkine receptor, so prime the microglia toward a more activated state, can we drive the depletion forward? And the answer is that we can. So in this mouse, both male and female decrease in their remaining microglia, so increase in their depletion efficacy. Um, I did not do TREM2 knockout, CX3, CR1 knockout, which would be very important because you would fully drive these cells toward a more activated state, but crossing those is just a pain. And there's just not enough time to cross these two knockout lines together and to generate some outlines. So I stopped here, but we expected that if we combine TREM2 knockout with a CH3 CR1 knockout, so microglia that lack TREM2, so lack the surviving pathway, but if we inducing a prime state of microglia, we can then drive them to depletion way faster. And I think that's it. Yeah, that's everything. We found that the same PLX treatment resulted in 60% brain-wide depletion in microglia male versus 80%, yeah, in females versus 80% in male. We found that PLX shifted male and female microglia toward a reactive and phagocytic phenotype, but these phenotypes are different in two sexes, in which microglia remaining after treatment in females upregulated expression of genes related to proteostasis and autophagy, so genes related to maintaining their population, while those in the males upregulated their mitobiogenesis, especially complex 1 subunits, driving more toward inflammations and phagocytosis. We surprisingly found that TREM2 knockout did not promote microglia ablation, but CH3CR1 deficiency does facilitate microglia removal by PLX in females, which then we conclude that because of this, we think that the basal reactivity of microglia is what governs their susceptibility to CSF1R inhibition. I feel like this research does not make a lot of sense if you actually not studying microglia for a living, because the question would immediately be, well, why you care? But male, female, or sex-dependent dimorphisms in disease prevalence as well as in cell types I start to attract a lot of attentions. And because microglia is starting to positions themselves at the heart of driving a lot of neurodegeneratives and neurological diseases, understanding how they functions and their survival mechanisms is crucial to understanding how to target them in male versus females. Just like getting the drug doses right, because as we see, if we use the same dosage, the female microglia still remain 100%, and we might even get the disease worse, right? Because we actually don't know their phenotypes after they've been with the drug for too long. So those are kind of the ramifications of this research. Another thing is that PLX get used a lot in microglia research. And usually when people used it and when they wanted to get rid of all microglia, they just try to hit it with even a bigger hammer and treating the microglia for even a longer amount of time. So I'm treating them all here in like a standard amount of time, which is about 2 weeks. But a lot of labs starting to treat them for like a whole month or like 3 months period, which then you will cause change to a lot of the micro environment in the brain without understanding if there are inherent change at the beginning in this microglia. So with that, I would like to acknowledge my previous labs and all the people that make this research happen, as well as fundings. And any questions? Thank you.
Yeah, thank you so much for your excellent talk, Lynn. And so now with the Q&A, so I will monitor. So if you have any questions, just feel free to raise your hands, and or if you feel like you want me to read the question, then just put it in the chat. So we'll start with a few questions from my side. So Lynn, I'm just curious about the level of the CSF1R expression
Mm.
in male versus female microglia, like before the treatment. Is there any difference between the two sexes?
Oh, the level of CSF1R expression at baseline between male and female, you want to ask if there's any differences in them? Yeah, so at baseline alone, there are already slightly higher CSF1R in male versus female. So not in the direction that we expect, but we think that because there are the CSF1R in males might be more sensitive to being depleted compared to the CSF1R in males are more sensitive to the inhibitors compared to female, but there are more receptors in male compared to female. So a
So
little
does
bit counterintuitive because you would expect that
mhm,
the female might express more, right? So
mhm.
the male expressed way more CSF1R, but then got depleted a lot more, and their protein level of CSF1R also decreased a lot more pronounced compared to female.
Yeah, this raises the question whether there is a sex differences in the binding affinity of the receptors. For
Yeah.
example, like the female receptors may have like higher binding affinity compared to the male.
Yeah,
But
that
yeah, that's
yeah. No, 100% agree. That's what we kind of think, but like we just don't have the tools to pursue that path
mhm.
further. And also I need to graduate. Just
Yeah, that's
that's
true.
a more way more
Yeah.
important.
Uh,
Yeah, the graduation is factoring in a lot in how we carry
mhm.
this forward. But yeah,
Yeah.
100%. Yeah.
Yeah, and also, because you see the galectin 3 positive cells are more
Mm-hmm.
resistant to the CSF1R inhibition. So I just
Mm-hmm.
wonder because galectin 3 is involved in cell adhesions. Is there a
Mm-hmm.
chance that what do you think about the impact of CSF1R inhibition on the niche? Whether the cells that interact more with the galectin 3 positive microglia have an impact on the cells next to it. So that you have some kind of like a niche effect to protect the microglia.
Yeah, no, definitely. So, well, because you, tại vì anh cũng làm về immune cells ấy. Yeah, it is a protein that just bind beta galactosidase. Nó cũng là một cái well-known protein in just inflammation pathway in general. So we do expect some non-cell autonomous
Mhm.
effects by just killing these cells with CSF1R. And the reason that the galactin-3 positive microglia survive may be also because of the niche difference. Another thing that I didn't mention is that um, at the beginning, I show Alzheimer's disease related microglia that are very resistant to being depleted by CSF1R. Aside from the fact that these cells start to express really low level CSF1R because of the aging process, they also express a lot more galactin-3. So galactin-3 also attract a lot more attention because it's get upregulated a lot in neurodegenerative diseases. Um, Another interesting thing is that it's also upregulated in developmental microglia. So not the adult microglia, but embryonic microglia also express really high level of galactin-3. Um, so there is this kind of comparison between developmental versus neurodegeneration. Are they just, like going back to developmental phenotypes and then just get activated at the wrong time and stuck chronically in that state? Those are all all valid questions. But yeah, I don't have the
Mhm.
answer to that, but I think yes. Yeah.
Mhm. Yeah.
Because it's not like they just like randomly one there, one here. They usually, if there's some galactin-3 positive, they kind of cluster together. So I would guess that.
Mhm. Yeah. And I guess the final question is, when you have the CSF1R inhibition,
Hmm.
you saw the decrease in the galectin-3 expression. I mean, like the protein level. What do you have any like? Do you have any thought on like why the inhibition of the CSF1R receptors affect the protein level of the galectin-3? And also, the galectin-3 has like protective effect against the CSF1R inhibition. I kind of like kind of different when I think about like two events.
Decreasing galactin-3. Galactin-3 got increased with PLX
That
treatment.
increase. Oh, okay. So
Yeah.
probably I misunderstood.
Yeah,
This.
yeah. So what we think is that at baseline, there's already a population. So the thing is that this PLX drug is have a very titratable kind of effect. So the higher the drug dosage and the longer period that you sit this animal on drugs, eventually the microglia will get wiped out. And the interesting thing is that they actually never get wiped out 100%. So that is what I find really intriguing because you can see this microglia on this drug for months and there's
Hmm.
still a very tiny population of them remain. So there's this like subventricular population of microglia that still remains after a prolonged treatment of PLX at really high dosage. If we if you use like a bunch of like if you really hammer it hard enough, they will also go. But that like very lethal though. Like that's you kill basically all macrophages in the body as well to
Hmm.
get rid of these microglia. But they have this very titratable. Um, effects. So I feel like, yeah, there are many subpopulations of microglia that have different susceptibility to being depleted, first of all. And they also might spread, not only galectin, but I feel like a galectin is just like a marker that is a part of these microglia heterogeneous phenotypes that exist in different brain regions. And maybe also associated with different types of neurons because neurons glia interactions and matching also start to emerge as a very prominent field, I guess, an area of research. People start to find out that, oh, microglia is not only regionally heterogeneous, but they also match with the neurons that they interact with in that microcircuits they're in. So there are a lot there that need to be
Hmm.
unpacked, which complicated the problem more, I think.
Hmm. Yeah. Um, I guess people don't really have any question.
Mm,
I have like one last question, Lynn. Um,
yep.
I kind of I kind of probably I missed the beginning, but why do
Um,
you want to inhibit the CSF1R signaling in the microglia?
Yeah. So the thing is that so at the beginning, this research spun from like very serendipitous finding. So when I was doing Alzheimer research, I tried to get rid of microglia for another project, and I can't. That's
Hmm.
basically it because they're too old, and I tried to treat them with a drug. We thought the drug got expired, and we trained vendor, and they're still there. So. That's when I actually have to go back to the literature and look at these and see, well, how do people actually get rid of them? And I found out that like it's kind of like buried in papers when you read and you're like, oh, they get rid of that, but then they never tell you how long they seed the mice on it and then what percentage of cells are actually gone and whether or not they actually just use male or female or like both sexes. And I found
Hmm.
out that people kind of realize that there is a difference, but like no one seems to care enough to make it like a deal and they just try to circumvent the problem by just hitting harder with the inhibition. And we thought it's very interesting because it's a very, very commonly used drugs in microglia research, especially
Hmm.
as microglia start to be studied in neurological and neurodegenerative diseases. So, like Alzheimer's, Parkinson's, and fetal alcohol syndrome, for example, or Fragile X syndromes. Those, for example, start to get a lot of microglia depletion therapies because people think that, oh, because it's sitting this is chronic inflammation environment, so if we just get rid of the microglia, we'll fix the problem.
Hmm.
But deciding what dosage to use and how long you see this Maya on this drug make a huge difference because you all know that CSF1R is also it's called colony stimulation factor for myeloid cells. So it's for every cells, right? The macrophages also get wiped out. Though at least there's some macrophages got wiped out. A lot of the monocytes also got wiped out. So how long the mice get stayed on the drugs make a huge difference. And at first we just interested in that. Like we literally just want to make a very small paper on like, oh, they're different. But then we think more on the line of like, what if they utilize different survival mechanisms at first? And what's the difference in male versus female microglia? Because we know that is the base, it might be the basis for a lot of the sex dependent divergence in disease prevalence. Um, for example, a lot of the autoimmune disease, including multiple sclerosis, are more prevalent in female. But some of the diseases are more prevalent in males that we don't know the reason, like Parkinson. Why that one suddenly switch when Alzheimer's are also more prevalent in females, despite
Mhm.
the regardless of the fact that female live longer. I think that myth
Mhm.
is got like chucked. But yeah, no, we basically just stumble upon the finding and then try to figure out if their survival mechanism differed. And we started really get interested in how they difference in metabolisms and stuff like that. The lab is carrying out that research, but I need to get out. So I didn't
Mhm.
get involved in that. But the lab is looking further into sex differences and metabolism differences and looking at in like other contexts as well, like in other diseases and in toxin exposure.
Yeah, thank you, Linh. Well, we have one question from Linh Nguyễn. Do you want to unmute and ask the question, Linh? Well, I guess I can ask the question for Linh. So she said she missed the talk, so she just looked at the paper and she asked whether you did you do the titration before deciding to treat the mice for 7 days only?
Do the titration in, oh, so the 7 days. Yeah, so the 7 day is important. I didn't mention this because I don't want to complicate it further, but we only treat it for 7 days for the flow cytometry experiment to get the microglia out. So that's a caveat with this paper as well, because all the immunofluorescence analysis we looked at 2 weeks because that is kind of what standardly used in the field. So that's what people use to get rid of like 80 to 90% of male microglia in general. The reason we only do it for 7 days because we want to actually get microglia. So microglia is already, so it looks like a lot in the image, but they're only 5 to 10% of the population in the brain. So in the way that we collect these data, and because of the constraints of like how long they can sit on the ice and the fax machine in general, we cannot let them wait for too long. So to collect microglia, we usually aim for at least a few thousand cells to be good enough for sequencing. And because of that, we can only treat them for 7 days. It's not like we want to only treat them for 7 days. Like ideally, we want to do everything the same way, and can compare these microglia directly. Because this is true. Like if we only treat it for 7 days, there's going to be differences in our sequencing data versus the actual IHC data, the immunofluorescent data. But if we treat for longer, we cannot like get these microglia, especially in male. There will be, I tried once. I think there was like a couple hundreds. So, which is very depressing. And the cells are not that healthy after fax. And so, yeah, that's mainly
Mhm.
why. And then someone asked, if I can read the question. Do they observe sex-dependent differences in microglia's genes expression, particularly the upregulation of immune-related pathways in male, come from chromosomal differences, or are they driven by hormonal regulation? This is such a good question. Um, I can say that it might come from inherent difference in sex instead of hormonal regulation. Because I know people have tried to look at females at different point in their estrus cycles and don't see differences there. There's also a paper coming out of Lee Lab on the four core genotypes. So male that have ovaries and female that have testes, if I remember correctly, that on the line of suggesting that it's kind of the interactions of the actual sex itself and the gonads that create differences, but hormonal, in terms of like variation in the estrous cycles of female doesn't make a difference. At least that's what I found out. But yeah, a lot of people start to look at genes versus hormones in male versus female. Another thing I would say is that a lot of this get primed by developmental. So unless you really detangle that, it's become hard to know. Um, because androgen signaling in during developmental and critical period is crucial to establish this microglia. The other reason why people don't care so much about adult microglia sex differences is because they look very similar in morphology and numbers, versus during developmental, there are actual more visible difference in male versus female in terms of like densities and stuff. So when they reach adulthood, it's generally regarded as, oh, they're like similar. They just microglia in the brain, and unless there's disease or something, they all the same. Um, and it's slowly getting recognized that that's not true. Their differences maintain until adulthood. It's not just developmental. And people also start to look at how the differences in disease as well. So yeah, that's a great question.
Mhm. Yep. Um, is there any question from the audience? I guess people are really saturated from the talk. Let's see. Tan has a question about what is the limitation of your research?
Yeah. So I feel like there are a lot because there's a lot that we didn't get to do. The first thing that you already point out is that we didn't do pharmacokinetics. So we don't know for sure. So we try, but we don't know for sure if, regardless of the index of PLX, which is similar between sexes, if we see any differences in sex-specific alternations of PLX entry into the brain and its metabolism, which might be different, we just didn't catch it with our one time point measure of PLX concentration. We also did not address any sex-dependent differences in their response to the CSF1R inhibition, whether it's cell autonomous or shaped by the environment, as you correctly point out. So that we did not address in this study as well. We also did not look further into their metabolism, although I would argue that take a lot more time. So that is definitely beyond the scope of this study. Yeah.
Yeah. Thanks again, Lynn, for your excellent talk, and we really enjoyed your science, and I think uh, everyone really appreciate the lecture. And with that, I guess we will conclude the webinar today, and I will see you everyone in the next events of IBSG Journal Club. And thank you, Lynn, and have a wonderful weekend. Yeah, bye
Yep.
bye.
Ô anh Vũ ơi, em bảo cái em nhớ cái ông thầy mà em nhớ ra là cái bác trẻ trẻ ấy, cái chú trẻ trẻ ấy, là Gabriel Victoria anh ạ. Anh có xem lab đấy không? Cái lab mà làm về lymphocyte dynamics với cả homing ấy anh. Em thấy chú đấy làm hay lắm, mà chú đấy cũng có mặc author grant. Lần gần nhất mà em nghe chú ấy talk thì thấy cũng đang tìm postdoc đấy, anh có thể check out thử.
Ừ, cũng ở Rockefeller. Cảm ơn em.
Đúng rồi, cũng ở Rockefeller luôn á. Tại vì anh
Ừ,
nhắc Rockefeller em mới nhớ ra là chủ đấy làm cũng relate những cái thứ mà mọi người đang làm, với cả em thấy cũng rất là kiểu interesting ấy. Hay vãi,
ừ,
kiểu em ít khi làm về peripheral immune cells nên là
ừ. Yeah, cảm ơn Linh.
anh tưởng